Bismillahirrahmanirrahim. Welcome again, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video we are going to talk about 3' polyadenylation. In previous video lecture, if you guys remember, we already talked about the 5' capping. We talked the introduction, we talked the mechanisms, we talked the requirements, and we talked the importance of the 5' capping. Today in this video we are going to talk about the 3' polyadenylation. We are going to talk the introduction, we are going to talk the requirements, we are going to talk the mechanism, and we are also going to talk about the importance of the 3' polyadenylation. 3' polyadenylation. Okay, now what does 3' polyadenylation means? As the name indicates, 3', when adenine residues or adenine nucleotide or adenine bases are keep adding by polymerase enzyme, when they keep adding adenine nucleotide after the transcription, and when we get a long stretch of adenine at the 3' end, 200 to 250, then this process is called as 3' polyadenylation, because poly means many, adenine means adenine, and lation means addition of adenine, the process through which the adenine is added. Okay, if we talked about the simple idea of the polyadenylation, so let's suppose this is the DNA, as you can see. This is the RNA polymerase, and this red one color is the messenger RNA. So remember, in the previous video lecture we already talked that 5' area is already methylated, or 5' area is already capped, as you can see. So capping should occur at the 5' area. Before 3' polyadenylation, the capping should occur, and we call it 5' capping. So we already talked about that. If you missed that video lecture, it is present into my channel, so go back and watch it. Now 60 to 50 nucleotide addition, then 5' capping occur. After that, the RNA polymerase will encounter some kinds of the signal, which we call the termination signal, and they will encounter termination sequence. Due to this way, they will activate some kinds of protein which we call it cleavage factor and cleavage proteins. Due to this way, the cleavage protein will cleave the RNA, and we get the RNA like that, as you can see. Then polymerase enzyme will keep adding adenine. So 200 adenine or 250 adenine, it will keep adding. It depends upon the microorganisms, that what kinds of microbes we have. In some microorganisms we have 200, in some microorganisms we have 150, in some microorganisms we have 250, like that. So after that, we have some other proteins which we call a PABP, polyadenylated binding protein, will bind to this specific area, and it will make the RNA structure in a circular form. So after that, the RNA become a circular in form. So this is a little bit idea about the 3' polyadenylation. Now, what are the main proteins factors that are needed for the 3' polyadenylation? For the 3' polyadenylation, we need adenine, nucleotides first. Second, we need RNA polymerase. Third, we need cleavage factor 1 and 2. Fourth, we need cleavage polymerase specific factor. Next, fifth, we need cleavage stimulatory factor, and next we need polyadenylated binding protein. So these are the main important things that we need for 3' polyadenylation. Okay, So how exactly 3' 3' polyadenylation will occur? So let's talk the mechanism. You know what actually happened? We have the DNA. Let me draw here. So let's suppose this is a DNA molecule, as you can see. At that specific area, during termination of DNA, if you don't know termination of DNA transcription, so I will recommend you that go back to my channel and watch the DNA transcription termination. What actually happened in termination of DNA transcription? At that specific area, the RNA polymerase reaches, and at that specific area there is the termination sequences like that. We means like that, TA, T, and that sequences. These are the termination sequences, and after that the RNA polymerase will add what we call that, it keep adding a termination signal like that, A, and what we get the structure like that. So remember, at that specific area the termination signal are present like A, U, A, and after the addition of some nucleotide, we have some other nucleotide which we call that CA. And after that, after further downstream of this RNA, we have some other nucleotide like GU. Okay. Now, once it reaches at that specific area, then all of you better know, with this RNA polymerase they have C terminal, and with this C terminal we have the two proteins, and these two proteins are the CSTF and CPSF. So what actually happen? These two proteins are get activated, and once it get activated, so first of all this CPSF bind to this area. So let me draw here. So this is C, let me write CPSF. It will bind to this termination signal, and after that, so this is another cleavage factor which we call that CSTF. It will bind at that specific area. Due to addition of these two cleavage factor, then some other cleavage factor is activated. Due to this cleavage stimulatory factor, it will stimulate some other cleavage factors, which we call it cleavage factor 1 and 2. So cleavage factor 1 and 2 are get activated, and it will bind to that specific area, as you can see. Sorry, not at that specific area. It will bind to that area. So this is the cleavage factor 1 and 2. This cleavage factor 1 and 2 make a cut in the RNA strand. So it will cleave the RNA after the CA. So just after the CA, it will make a cut in the RNA strand, and after that we get the RNA strand, as you can see. So at that specific area, capping is already done, which we call that 5' capping. It is already done. At that specific area we have the same things. We have the termination signal AA, U, A, and after some area, means after that we have CA. So CA is present at that specific area. Now it is cleaved, as you can see, and you know this is the complex. Remember, we have RNA polymerase, cleavage factor 1, 2, CSTF, CPSF. This is the complex formed, and after that we get the RNA in this form, and again the same protein are also binded with it, as you can see. This is the CPSF, and here we have the CSTF. Now once it binded here, then what actually happened? RNA polymerase will keep adding adenine at that specific area. So it will keep adding adenine. So almost 200 adenine it will keep adding. So when it reaches 200 adenine, so then there is conformational changes occur in the RNA polymerase, due to which it will lose its ability to bind, and then it will fall down. Then we get the RNA structure, as you can see. And after some time, these two cleavage factor are also released out, and we get the RNA like in this form. So 200. And we have also, so here I'm not writing again the termination. So we have also termination signal, as well as we have also CA adenine or nucleotide. CA nucleotide are also present here. And this is the 5' capping area. Now once the cleavage factors are released out from that specific area, then what happen? This polyadenylation binding protein will comes and they will bind to that specific area. So it will bind at that specific area. So we have one polyadenylation binding protein, which is a very huge structure, and it will bind at that specific area. And what actually it will doing? It will bring this 3' area as well as 5' areas closely to each other, as you can see. So this is the 5' area and this is the 3' area. So 3' and 3' polyadenylation area or polyatel and 5' capping are come closer to each other by this PABP protein. And when it come closer to each other, so we get a circular RNA like in this form. Here is the adenine. As you can see, and this is the capping, and this is the PABP or polyadenylated binding protein are bind here, and it will make the RNA structure like that. So circular RNA we get. This is the circular RNA. Okay. This is all about the mechanism of this 3' polyadenylation, that how it will form. After that we get the structure, the secondary structure of the RNA like that. So now, why this 3' polyadenylation is important? Now why it is important? The 3' polyadenylation is important for the exportation of this mRNA. It is important in exportation. What does it mean? It means that due to this 3' polyadenylation, the messenger RNA are easily be exported from the nucleus to the cytoplasm. First. Second is translation. Due to this 3' polyadenylation, the messenger RNA are get matured, and due to this maturation is ready for the translation process. Third thing is stability. So it will stable the mRNA highly. Due to this stability, due to this maintenance of the stability, once the mRNA becomes stable, remember, once the mRNA becomes stable, so it will keep the lifetime, or it will increase the lifespan of this mRNA. That's why polyadenylation is very important. Another importance is that, which is very main property, we call that exonuclease degradation. Exonuclease. What does it mean? It means due to this 3' polyadenylation, the exonuclease, which is an enzyme which can cleave the terminal end of the RNA. Remember, if the terminal end of the RNA like 5' area is already capped, so exonuclease will not cleave it. If the terminal area of the RNA, which we call it 3' area, is already polyadenylated, so it have no ability to cleave it. Remember, this adenine area is the junk portion. It doesn't participate in the protein synthesis, so it don't take any part in the protein synthesis, so it is a junk portion. We don't need it. So let's suppose the exonuclease comes and they will cleave. Remember, exonuclease have the ability to cleave one of the nucleotide at a time. So when it keep degrading, or it remove one of the nucleotide at a time, so then it don't have the ability to remove all the nucleotide. It will just remove a few nucleotide. Let's suppose 5 to 10 nucleotide it will cleave out, but the remaining nucleotide, adenine nucleotide, are already present. So it means these areas are not needed. So we can say, due to this 3' polyadenylation, we have also protect the mRNA from the exonuclease degradation. That's why 3' polyadenylation is also important. So this is all about the introduction requirements, mechanisms, and importance of the 3' polyadenylation. Hopefully you will get idea about the 3' polyadenylation, and if still you have any kind of confusion and question, you can share your question and confusion through a comment. I will try my best to give you answer as soon as possible. So if you like this video, make sure to hit the like button, share this video, and also subscribe the channel, and stay tuned, because in the next video lecture we are going to talk about the splicing. After the 5' capping and 3' polyadenylation, this RNA will proceed into the next stage, which we call it splicing. We will explain it in a detail in our next video lecture. Thank you so much for watching this video.